Welcome back to AllAboutRx. Today, we're diving deep into the microscopic communication hubs of our nervous system by exploring synapses and neuronal integration. We're going to explore how your brain takes these tiny chemical signals and integrates them to form literally every thought, feeling, and action you experience.
Now, to really set the stage for how monumental this is, I want you to think about this staggering number. 100 trillion. That's 10 to the 14th power. That is the estimated number of synapses actively firing and communicating inside your human brain right at this very second. It's kind of crazy, right? It's a network vastly more complex than the entire global internet, sitting right there inside your skull.
So here's our roadmap for today. We'll hit the anatomy of a synapse, the step-by-step mechanism, EPSPs versus IPSPs, synaptic integration and summation, clearing the synaptic cleft, and finally, neural pathways and networks. Let's get into it.
Section 1. Anatomy of a synapse, the microscopic junction. Okay, let's dive into this. A synapse is, at its core, a strictly one-way functional junction between two neurons. You've got the presynaptic neuron, that's our sender, and the postsynaptic neuron, our receiver. And the scale of this whole setup is just mind-blowing. A single postsynaptic neuron doesn't just listen to one neighbor. No way. It can receive up to 100,000 individual synaptic inputs from thousands of converging presynaptic axon terminals. Imagine a massive conference call where one person is simultaneously receiving messages from 100,000 different people.
Now, let's zoom in even closer on just one of those connections. You can see the synaptic knob, which is that swelling at the tip of the sending neuron. Inside it are these synaptic vesicles. Think of these like tiny molecular shipping containers, and they are fully loaded with chemical messengers called neurotransmitters. Between the sender and the receiver is the synaptic cleft. Now, here's an absolutely crucial detail. This cleft is a fluid-filled gap. It is physically too wide to allow an electrical current to just jump across. So, communication across this microscopic chasm has to shift from being strictly electrical to chemical.
Section 2. The step-by-step mechanism of transmission. It all happens in a remarkably precise 5-step sequence. First up, an electrical action potential travels down the presynaptic axon terminal and arrives at the junction. This electrical shift triggers step 2. Voltage-gated calcium channels open up, causing a massive influx of calcium ions into the synaptic knob. This calcium acts like a molecular key, which drives step 3, exocytosis. The synaptic vesicles fuse with the membrane and dump their neurotransmitters right into the synaptic cleft. In step 4, these chemical messengers swiftly diffuse across that narrow gap. And finally, step 5, they bind to specific receptor channels on the subsynaptic membrane, acting exactly like keys and locks to open up new ion channels.
Section 3. EPSPs versus IPSPs, the two fates of a synapse.
So the message has arrived, but what does it actually say? Well, you can think of the receiving postsynaptic neuron as this highly advanced biological calculator that has to interpret these chemical messages. Depending on exactly which specific ion channels are opened, the synapse will have one of two fates. It's either going to excite the neuron or inhibit it. An excitatory postsynaptic potential, or EPSP, happens when nonspecific cation channels open. A net positive charge enters the cell, causing a small depolarization, bringing the membrane closer to its firing threshold. On the flip side, we have an inhibitory postsynaptic potential, or IPSP, which happens through potassium or chloride channels opening. This increases the net negative charge inside the cell, causing a hyperpolarization that pushes the cell further away from its firing threshold. It's basically a microscopic game of go versus stop.
And this brilliantly illustrates the difference. Looking at the traces here, the EPSP creates a slight upward bump in the membrane potential, moving it closer towards that critical threshold. Conversely, the IPSP causes a downward dip, making it actively harder for the neuron to fire.
Section 4. Synaptic Integration and Summation.
Here's the thing. A single subthreshold potential, like one EPSP or IPSP, rarely reaches threshold on its own. One active excitatory synapse just isn't enough to trigger an action potential. The neuron actually requires a grand postsynaptic potential. That's a composite tally of all the excitatory and inhibitory inputs happening at the same time, all working together to finally cross the threshold and fire its own signal.
Now, what's really interesting about this is how that biological math actually plays out. There are three main ways. First is temporal summation. This comes from rapid, successive firing, where a single presynaptic neuron fires repetitively. The EPSPs piggyback on top of each other before the first one can even fade, stair-stepping their way up to the threshold. Second is spatial summation. This happens from simultaneous multiple inputs. Their individual EPSPs add together all at once to hit the threshold. And finally, we have EPSP-IPSP cancellation, where an excitatory go signal and an inhibitory stop signal fire at the exact same time, effectively canceling each other out and keeping the neuron at rest.
To ground this microscopic math in everyday reality, let's look at a reflex we all know: bladder control. When your bladder stretches, it sends reflex EPSPs to the spinal cord. But usually, that's not enough to trigger urination on its own. When you decide it's an appropriate time, your higher brain centers add voluntary EPSPs. Through spatial summation, they combine, hit the threshold, and, well, urination occurs. Conversely, if you need to hold it, your brain sends voluntary IPSPs down the line. These can actually cancel out the reflex EPSPs from the stretched bladder, keeping you comfortable until you can find a restroom. It is literally biological calculus happening inside you in real time.
But this perfectly timed system begs a really important question. Once that precise chemical message is delivered and the math is done, what happens to all those leftover chemical messengers floating around in the synaptic cleft?
Section 5. Clearing the synaptic cleft.
So to ensure the system is ready for the next signal, the body has to meticulously clean up the synapse. If it didn't, the receiver would just keep firing endlessly. To wipe the postsynaptic slate clean, our nervous system utilizes 3 rapid clearance mechanisms. First up is simple diffusion, where neurotransmitters passively drift away from the narrow gap. Second is enzymatic degradation, where specific enzymes act like molecular scissors to chop up and deactivate the messengers. And the third is reuptake transporters, where active transport pumps vacuum the neurotransmitters right back into the presynaptic terminal to be recycled.
The crucial point here is that these clearance mechanisms are so vital that they're actually heavily targeted by clinical therapeutics and drugs of abuse. Take SSRIs, like Prozac or Paxil. They selectively block the reuptake transporters for serotonin. By preventing the cleanup, serotonin lingers longer in the synapse, prolonging its mood-regulating effects, which is great for treating depression. But on the darker side, we have cocaine's impact on dopamine pathways. Cocaine competitively blocks dopamine reuptake. This traps dopamine in the synaptic clefts of the brain's pleasure centers, leading to prolonged overstimulation and ultimately a devastating cycle of addiction.
Section 6. Neural pathways and networks.
Let's zoom out for a second. Single synapses don't exist in isolation. They're part of a vast, interconnected web defined by 2 main architectures: convergence and divergence. Convergence is when thousands of presynaptic cells all channel their inputs down into a single postsynaptic central hub. It's how your brain synthesizes multiple different sensory inputs into one cohesive concept. Divergence is the exact opposite. A single presynaptic neuron branches out to simultaneously influence multiple downstream postsynaptic neurons. Every single neuron is basically acting as both a receiver of converging information and a distributor of diverging information all at once.
It really makes you sit back in awe of the machinery inside our heads. If every thought, emotion, and action you take is ultimately just a grand calculation of microscopic chemical votes, excitatory and inhibitory, how does this invisible tallying actively shape your conscious reality? It's a profound mystery. Hope you understand the concept, and like, subscribe, and comment. See you in the next explainer.